Hi guys, welcome to the Unit 3 review, Cell Biology. Of course, you might ask yourself, why am I looking at this picture of the Milky Way galaxy if we're talking about cells? And I just wanted to remind you of the immense complexity that makes up living systems. Remember, the Milky Way has about 200 billion stars, and you've got a number of cells that's a couple orders of magnitude greater. Remember that you start off as one cell, or actually, more accurately, two cells, the sperm and the egg, and they come together in fertilization to form a zygote. And that zygote begins to undergo cell division, replication, in a process called gastrulation. Notice the cells don't come apart, they stay together in a ball. And eventually, 9 months later, you got a human. Initially, all these cells are identical, but over time, those cells undergo cell differentiation. They become different parts, and eventually you end up with this complex organism. You have about 70 trillion cells. It's a very, very large number. But those cells aren't the same. They're organized into all kinds of different structures. This is a microscopic organism, and it's actually one of the organisms that was initially termed an animalcule. Today we call it a protozoan or a zooplankton. It's an animal-like protist, or I'm sorry, an animal-like animal. This is an animal here. This is a invertebrate, microinvertebrate, and you find these guys everywhere. In fact, we found a couple of these in our pond water. It's a rotifer, and he's got all kinds of complex structures internally. In fact, that little red spot there's an eye spot. It's a light-sensitive organ. So in this unit, we'll find out what are our cells, specifically how big they are, and what are they made of. What's the organization that we find there? And the guys who originally investigated cells are in a short list here. So first and foremost was Robert Hooke, and Robert Hooke is an Englishman in the 1600s in England. He did a lot of stuff, but one of the things he did was build a microscope, and he looked at something called cork cambium, which is part of the bark that you make corks out of. And he noted that the structures there looked a lot like the places where the monks lived in the monastery down the street. They called them cells. And that's where we get the term today. That's a picture of Robert Hooke's microscope, and that's a micrograph of cork cambium with the little cells there present. Next in line was Antoine van Leeuwenhoek. He's Dutch, and a brilliant guy. Remember, the Dutch were very powerful naval presence, and of course, if you are on a ship, telescopes are important. So Antoine was really interested in building lenses, and when he built his microscope, he saw these, hordes of little tiny microscopic creatures that he coined animalcules. In fact, von Leeuwenhoek's considered the father of microbiology. That's the study of microscopic organisms. Next in line was Matthias Schleiden. He's from Germany. Like Hooke, he looked at plants, and he finally said that all plants are made of cells, individual cells. And his buddy Theodor Schwann noted that all animals are made of cells. Eventually, there was a guy named Rudolf Virchow who, I guess, said ironically that all cells come from pre-existing cells, which begs the question: which came first, the chicken or the egg? Anyway, Rudy came up with the cell theory. So Rudolf Virchow postulated the cell theory, and he said that all organisms are composed of one or more cells, that the cell is the basic unit of living things, and that all cells arise from other cells. This is a ciliate. I want to say this is a paramecium, although I'm not positive, but it's a ciliate. It's a protozoan. This is actually a unicellular eukaryote. It's a protist. It's a protistan organism, and remember, it's eukaryotic. So in our class, we study microorganisms using the compound light microscope. It's a light microscope because in order to use this thing, you have to be able to pass light through the object. So I can look at transparent objects, I can look at translucent objects, but I cannot observe opaque objects with the compound light microscope. It's a compound microscope because there's always two lenses in series. At the top near your eye is the eyepiece lens, or ocular lens, with a magnification typically of 10x. And then down bottom you have the ability to select multiple objective lenses. On our scopes we have 3 objective lenses, a 4x, 10x, and 40x lens. And we can magnify objects by lining up those lenses in series And if I use the 4x objective lens and the 10x ocular or eyepiece lens, I get a total magnification of 40x. There's another feature of microscopes, or a measure of microscopic ability, and that's called resolution, and that's the lens's ability to discriminate 2 points. And you can imagine if I drew 2 tiny points close to one another up on the board, like we did in class, if you got far enough away, you wouldn't be able to tell whether it was 1 point or 2 points, and that distance and your ability to discriminate is called resolution. Down at the bottom here, you see some cheek epithelial cells. We try to measure the diameter there, and hopefully you got a bead on that. Here's some rotifers down here that we've actually photographed in class. And then to the right, I have a couple of amoeboid creatures there, and they're getting eaten. They didn't realize it yet, but in a sec they're going to be like, "Hey, what's up? They're fighting." Well, it doesn't matter. It's all over for you two. When it comes to cells, size does matter, and bigger isn't always better. In fact, quite the opposite. And that has to do with something called the surface area to volume ratio. And if you see the illustration here, you can see that for a cell that had a 2, or for a cube that had a 2 by 2 dimension of inches, it would have a surface area of 24 square inches. So each face would be 2 times 2, or 4 square inches. We've got 6 faces, which gives us 24 square inches for the total surface area. To calculate volume, of course, you take the x, y, and z axis, so it's 2 times 2 times 2, which gives us 8. Now, when we divide surface area by volume for that 2-inch square, we get a surface area to volume ratio of 3. And when we move up to those larger cubes, that 8-inch square has a large surface area. 8 times 8 times 6 comes out to be 384 square inches. However, its volume increases exponentially, right? So its surface area is 384, but its volume's up to 512, and now its surface area to volume ratio is below 1, 0.75. And that little cell's got this ratio of 3, big cell, 7.5. That's bad. Why? Well, remember that cells need to, in order to survive and maintain homeostasis, they have to exchange materials with the surface. Not only that, but if they're too big, it takes a lot of energy to move stuff around the cell efficiently. So small is better when it comes to cells. And so here's some examples of some typical human cells. They vary in size and shape. We measure them in micrometers. And if you recollect, 1/10 of a millimeter is 100 micrometers. There's some sperm up there at the top. To the right is a diagram of an ovum, and then beneath that's a smooth muscle cell. To the left, you can barely see them, are the red blood cells and white blood cells. There's some big pile of red blood cells to the left. In the center is some cardiac muscle tissue. It's actually, I think it's in vitro. It's just grown in glass. In vitro means in glass. And you can see it's contracting. It does that by itself. To the right is adipose tissue. That's the fat that makes up your fat. And of course, we have what kind of fat inside of those adipose cells? Triacylglycerides. That's how you store energy. Down at the bottom, I've got a human embryo with some cool staining of their nervous system. In the center bottom, I see in vitro fertilization. That's an ovum there, and it's being injected using a capillary with DNA from some donor. So I have, a fertilization event right in front of us. And on the far right, I've got a nematode worm. That's a roundworm, and it's being attacked by a bunch of human white blood cells. And are you exposed to roundworms? Pinworms? Yeah, like 2/3 of all kids on the planet are exposed to pinworms, and you can tell by looking at them, or more explicitly what they're doing with their behavior. Which brings us to this big distinction of prokaryotes versus eukaryotes, okay? So remember, when we talk about cells, we talk about whether or not you're unicellular or multicellular. When you talk about living things, we broke them up into the 3 domains, archaea, bacteria, and then eukarya. Now, both archaea and the bacteria are prokaryotic organisms. And of course, the eukaryotes are all everybody else. So what does prokarya and eukarya mean? Well, prokaryote means before the kernel, and the kernel means the nucleus. Eukaryotes are new kernels. That is, when I look at a eukaryotic cell, I can see structures internally, but most importantly, I see that nucleus. All right, so the karya refers to the kernel, and we're talking about the nucleus, kernel of corn, if that helps you. So what is it that differentiates prokaryotes from eukaryotes? Prokaryotes are all bacteria. They're either archaebacteria or they're bacteria proper. eubacteria, they don't have any membrane-bound organelles. They're really small. They have DNA, but it's CCC DNA, covalently closed and circular. They have little circular pieces of DNA. And of course, they reproduce by binary fission, as you can see down here. And they can reproduce really quickly. They also have high mutation rates, which gives them some adaptive advantages. Eukaryotes, on the other hand, include all the kingdoms of eukarya, which are protista, fungi, plant, and animal. They have a nucleus and other membrane-bound organelles, and their DNA is organized into chromosomes. Well, let's look at prokaryotes in a little detail so we can compare them with the eukaryotes. Remember, prokaryotes are either archaea or bacteria, and of course, those are the domains, but the kingdom for archaea are called the archaebacteria, and the kingdoms for the eubacteria are the eubacteria. So archaea, remember we spoke about these guys. Archaea means old. These are ancient forms of life, and they include the methanogens, which may be the most common organism on the planet. They make methane, and then they eat methane. The sulfur bacteria, they make sulfur compounds and they eat sulfur compounds. And one of the features of the archaebacteria is they're all autotrophs. They make their own food, but they're chemoautotrophs. They use chemical processes to get that energy. And they're often called the extremophiles because they inhabit such extreme circumstances. So you find these guys buried miles deep in the rock or in ice or in extremely salty conditions, or even in hot acid. Remember, the thermoacidophiles love hot acid. You find them in deep sea vents. You even see them out in Yellowstone Park at those geysers. The archaebacteria are useful, though. You need them to help you digest your food. Eubacteria, on the other hand, are also helpful. We have lots of helpful eubacteria. E. coli in our gut helps us digest, but there are pathological forms of E. coli, or virulent forms. And we also have some of the eubacteria you might be familiar with include things like streptococcus. You might have strep throat, or staphylococcus. When you get an infection on your skin, that's a staph infection. And then there's a whole host of them, Clostridium, there's a lot of them. But they're also very helpful. So most of the photosynthesis on the planet is actually done by the cyanobacteria, which are marine protists or prokaryotes, and they really create most of the oxygen on the planet, reduce this and capture CO2. Again, they reproduce through binary fission. In binary fission, that circular DNA is replicated, and then the cell divides. Pretty straightforward and simple. So bacteria, simple, small, no membrane-bound organelles. They have some of the features of modern cells. They have a cell membrane, but it's a primitive cell membrane. They have ribosomes, but they're primitive ribosomes. And it's not within the scope of the course to discuss that. Of course, you're welcome to go check it out, what makes their membranes and ribosomes different. So there you go for prokaryotes. And of course, in a simple terms, we can compare them with the eukaryotes. Eukaryotes are complex. This is a Daphnia here. This is also a microinvertebrate. And she's checking out a volvox, which is actually a phytoplankton. So that's a unicellular eukaryotic protist that does photosynthesis, phytoplankton, volvox. And the key to understanding eukaryotes is complexity, right? When you look at a eukaryotic cell, you see a nucleus, you see other structures, and the rest of the unit is going to be devoted to understanding eukaryotic cell functions. That's it, and I'll see you on the other side.